Hello and welcome to Nikolay's genetics lessons, and today I have prepared very easy problem for you. Only one question: which of the following proteins do not have quaternary structure? And here is a variance of the answers. First, let me briefly remind you what kind of structures we can find in the proteins. And the first would be primary structure. This is just a sequence of the amino acids. Secondary structures like alpha helix and beta sheets would be made due to certain forces like hydrogen bonding and due to arrangement of the R groups of the amino acids. Some of them can be hydrophobic and some can be hydrophilic. And tertiary structure would be just arrangement of these two structures, alpha helix and beta pleated sheets, in certain three-dimensional form. Sometimes it can be active protein, but it have to be made from the single polypeptide chain. But sometimes, in order for this protein to be active, it have to arrange with other proteins. Sometimes it can be the same type of subunits that just aggregate together. Sometimes it can be different proteins that have to aggregate together in order for the final protein to be active. Now let's return to our question and variance of the answers. By the way, some of you may think, how I supposed to know all the proteins, their shapes, their names, if there are 23,000 genes and about 100,000 different proteins in our organism. You don't have to know all of them, you just have to know those ones that is used in textbooks as a model proteins, just like model organism we use in biology, for example, like laboratory mice. So all these proteins you just have to know their function, their structure, and you have to be able to recognize them. For example, answer A, P53 protein. What is important about this protein? Why we have to know it? It is very important protein which we call tumor suppressor protein. And here's a picture which represents this protein, and this double-stranded DNA is not part of this protein, but it shows how it binds, and it is made from one polypeptide chain, but it's only active when 4 subunits would arrange together, and in this form it would be active. It's called tumor suppressor protein, but actually this is misconception. It is actually activator protein. It activates another gene, which is P21, and that gene, when active, would make a protein that suppress cell division. P53 is not a protein of our choice. Next variant, DNA polymerase. Here on these pictures, it's going to be this protein, which, as you see, consists of many different subunits, and each subunit is made by expression of different gene, and then they would aggregate into this active protein, which function would be to make a new strand of the DNA when cell would divide, and DNA, each chromosome, also have to divide. So this is also not a protein of our choice. Now let's check next one, ribosome. This is also interesting variant, and everyone knows ribosome, and I hope you can recognize this protein. It is here. And everyone knows that ribosome consists of small subunit and large subunit. What is also interesting, that these two subunits is made between 40 and 80 different polypeptides. Polypeptide chains or proteins which aggregate together, and between 3 and 4 RNA. So you see RNA also shown here, and RNA here takes about 50% of the space. So this is very, very complex protein. And the function of the ribosome would be using messenger RNA to make a polypeptide chain, to make all the rest proteins. And this is not a protein of our choice. And next, our choice is going to be variant D, hemoglobin. Those people who subscribe to my channel know the structure of this protein very well. It is here. It is made of 2 alpha subunits and 2 beta subunits. And its function would be to carry oxygen molecules throughout our bodies from our lungs to our organs, and from organs and back to lungs, CO2. So this is also not a protein of our choice, and now we're left with only two proteins. And probably many of you know that this is two very small proteins. Which one to choose? One of the smallest proteins in our organism is insulin, and thus it is very small polypeptide. Actually, the structure is not very simple. It was the first polypeptide that has been sequenced, and here its structure. Actually, it's made of two chains, two polypeptide chains, which are connected by two disulfide bridges. Actually, there are three disulfide bridges, but one of them within the same polypeptide. So two polypeptide chains are held by two disulfide bridges, and thus it is expressed from the single gene, but when polypeptide chain would be made, it undergoes certain modifications and it would be divided to two polypeptide chain, which later also can aggregate in such complex molecule as shown here on the picture, which we call hexamer. And here in the center we can find zinc atom, which attracts and holds these three proteins together. Thus it is not necessary for this protein to be active. It can be active as a single protein, but still it consists of two chains. So it has quaternary structure, and this is not a protein of our choice. And as you see, we have only one variant left, myoglobin, and here is the structure of this protein. It is very simple protein. Actually, homology of this protein is over 90% to the beta unit of the hemoglobin. It also has heme group here with, as you see, oxygen molecule attached to it. But unlike hemoglobin, myoglobin doesn't have to aggregate into the complex structure, and it can perform its task, which is to supply organs and tissues with oxygen by itself. The difference between these two would be that this protein, hemoglobin, we can find in the blood, and this one we can find in the tissues, mostly in the muscles, where it stores oxygen and performs as backup for oxygen supply in high demand periods. So as you see, the only protein that doesn't have quaternary structure is going to be myoglobin, and this is going to be our choice. And this is all for today. Subscribe and see you in the next video. Goodbye.